Hello everyone, welcome to Positron Academy. In this video, we are going to delve into a slightly different topic, finding much relevance especially for the nuclear medicine students. So the topic is peptide receptor radionuclide therapy using lutetium-177 dotatate for neuroendocrine tumors. The purposes of this video are mainly to increase the understanding of the concepts of theragnostics and peptide receptor radionuclide therapy and as they apply to neuroendocrine tumors. Along with it, we will also be reviewing the phases 1, 2 and 3 of the clinical trial data which led to the approval of lutetium-177 dotatate. So this should foster the understanding of the practical aspects and the future directions of PRRT for NETTERs. So this therapy will continue to be studied and optimized. Let us begin by knowing about neuroendocrine tumors. So NETs are a heterogeneous group of rare neoplasms that originate from cells with a neuroendocrine phenotype. The unique characteristic of such tumors is the overexpression of somatostatin receptors called SSTRs, and the tumor occurrence is mainly distributed in the gastroenteropancreatic tract, which ranges from 72% to 75%, as well as in the bronchopulmonary system or the lungs, to be having a 25% occurrence. And NETs can be classified into functioning and non-functioning types, with the former, the functioning type, producing and releasing an excessive amount of peptide or amines into the system, which is in turn giving rise to symptoms like facial flushing, diarrhea, along with hypoglycemic state and gastric ulcers or even skin rash. But the latter, which is non-functioning type, doesn't involve in excess production or release, but tend to show general symptoms of metastasis such as weight loss, bleeding, and abdominal pain, which make prognosis tough. Moving forward to somatostatin. Somatostatin is a hormone produced by many tissues in the body, principally in the nervous and digestive systems, and it exerts an anti-secretory effect over the other hormones, along with having a growth inhibitory effect. So this oligopeptide somatostatin, it undergoes a rapid enzymatic degradation in blood. Hence, stabilized analogues of somatostatin, namely octreotide and lanreotide, are used to achieve a prolonged duration of action. So here we see the structures of somatostatin, octreotide and lanreotide. You would like to note that the circled amino acids over here are the essential amino acids which are responsible for receptor binding. Moving forward to somatostatin receptors, these are G protein coupled membrane bound receptors which occur in 5 subtypes in humans, and with the SSTR2 subtype 2 being being typically overexpressed in most differentiated NET cells. And the somatostatin, it exerts its action by inhibiting the G protein dependent cyclic monophosphate. Coming to the somatostatin receptor ligands, here is this chart. We see the available ligands. So for the purpose of PET imaging, which involves positron emission and subsequent detection of the annihilation photons, gallium-68 labeled SSTR ligands find diagnostic use. primarily due to the positron emission property of gallium-68, which is given in this chart. And for the purpose of therapy, that is in PRRT, we are introduced to two radionuclides called yttrium-90 and lutetium-177. Both emit electrons, with lutetium-177 also showing gamma emission properties. Here I would want to emphasize on the fact that beta emission finds use in therapeutic purposes. So if you see high dose therapy protocols, you would note the use of iodine-131, which is a beta emitter. I will be highlighting the mechanism of action in the coming slides, and I also want to bring to your attention to the linkers or chelators used, namely DOTA over here, with another example of a chelator being DTPA, and the targeting peptides, which are octreotide and octreotate, and together when chelated with DOTA, we get DOTA-TOC, DOTA-TATE, and even DOTA-NOC. So when a radionuclide such as gallium-68 or yttrium-90 or lutetium-177 is chelated to an appropriate targeting peptide, it gives us a radiochemical. This radiochemical, when is validated through preclinical and clinical trials, is released into the market as a radiopharmaceutical. Let me explain to you the concept of theragnostics before we enter into knowing about PRRT. As explained just now, a target molecule is labeled with a diagnostic radionuclide. This is used for quantitative imaging in PET or SPECT, and with the same targeted molecule, if we now tag a therapeutic radionuclide, we could administer the same to the patient in order to obtain a therapeutic effect, with tumoricidal radiation dose being delivered to the metastasis, and which can be viewed in via imaging, the response of the therapy. And in the case of NETs, the targeting moiety remains unchanged, with the diagnosis performed using gallium-68 and subsequent therapy carried out with replacement of gallium-68 with lutetium-177. So simply put, diagnostics and therapeutics clubbed together culminates to theragnostics. And the best example of a theranostic agent would be iodine-131, which is used for simultaneous imaging and therapy. So we can now define PRRT as a molecularly targeted radiation therapy composed of a beta-emitting radionuclide chelated to a target peptide, which is designed to target the specific receptors which are overexpressed on the tumors in order to deliver a cytotoxic radiation to these tumors. So PRRT is not specific for NETs, but can also be applicable for prostate cancers, where PSMA becomes the targeting moiety. So in order to determine which radionuclide will be the best choice, we must know their properties. So if you follow this comparison chart, it will be clear that since yttrium-90 is a long-range beta-emitting isotope having a maximum beta particle energy as 2.28 mega-electron-volt, it should have more penetration depths in tissues, and that is with the maximum penetration depth in soft tissues being 11 millimeter, and so it should subsequently be advantageous when dealing with larger tumors. Again, if we see lutetium-177 on the right hand side having a short range beta emission with a lower maximum beta particle energy being as 0.498 mega electron volt, and the maximum soft tissue penetration depth that is achieved is 1.77 1.7 millimeters. Mm, so it will be advantageous in dealing with small sized tumors, and very importantly for inoperable tumors, as lutetium 177 also has gamma emission lines, which is missing for the case of yttrium 90. We see the uh, uh, the the possibility of two gamma emission lines, which are 113 keV having a 6% relative abundance, and 208 keV having an 11% relative abundance. So this proves to be beneficial in obtaining post-treatment images using a gamma camera. I hope till now you have understood the concepts introduced to you. Here you see two widely used targeting peptides as somatostatin analogs called octreotide and octreotate. Octreotide is actually octreotide acid. So if you want to obtain octreotide from octreotate, then we just need to reduce this terminal threonine into its corresponding amino alcohol. And with the addition of the chelator DOTA to octreotide, it gives us DOTA TOC, while DOTA chelated to octreotate will yield DOTA TATE. So these are attached at the 0 positions. Now that the structures are clear, let us move on to knowing about their properties. DOTA behaves as a bifunctional chelator, meaning both its ends are able to carry out chelation, and DOTA TOC has been found to show a higher affinity for SSTR2 but lower affinities for SSTR5 and 3. So that is pretty impressive, while DOTA TATE has been found to show a 6 to 9 fold higher affinity for SSTR2 than DOTA TOC, but it has no affinity for SSTR5 and SSTR3. So non-specific binding is totally removed from as an option for DOTA TATE, while this should make one thing clear in front of us, that DOTA TATE is showing high specificity for SSTR2 over DOTA TOC, as well as showing no affinity whatsoever for any of the other receptor subtypes, which is pretty required property when we are moving for dealing with such inoperable tumors. Like, we want to be totally specific in order to operate only those tumors which are showing an overexpression of SSTR2 in these entities. Now, moving forward to Lutathera. Lutathera is actually the marketing name for the compound, which is radionuclide lutetium 177 and DOTA TATE, and it is special because it is the first FDA as well as EMA approved radiopharmaceutical for peptide receptor radionuclide therapy. FDA stands for US Food and Drug Administration, while EMA stands for European Medicines Agency. So Lutathera was approved by the EMA in 2017, followed by the FDA in 2018 for the treatment of SSTR positive gastroenteropancreatic neuroendocrine tumors. With the current situation being that it might be applicable for other tumors as well, like lung LEDs. So this here is the schematic of the radiopharmaceutical. We can see the radionuclide in red, and the chelator DOTA in green, and the octreotate, which is the rest. So lutetium is chelated to the DOTA, which is attached to the targeting peptide, together forming the radiochemical, which is lutetium 177 tutartate. I have provided to you the IUPAC name of the Lutathera, the compound. And so now let us move forward to the inclusion criteria. So it means that which patients should be considered for undergoing this therapy. Firstly, we should look into the grade. It should be at grade 1 or grade 2 of well differentiated NET, and the stage should be inoperable. I mean, the tumors should be inoperable or can be metastatic, with sufficient tumor uptake on diagnostic SSTR imaging. And coming to bone marrow and kidney, because since this therapy can cause nephrotoxicity and myelotoxicities to some extent, therefore we must make sure that the patient has sufficient bone marrow reserves with a creatinine clearance of more than 30 ml per minute. And the functioning, the Karnofsky performance status of the patient should be should be at least more than 50%, which denotes the activity status of the patient. And the prognosis is we have an expected survival of more than 3 months for these patients. And most importantly, we must obtain a signed informed consent. This here shows the lutetium dotatate administration. So this is showing us the gravity method. There are two methods of lutetium dotatate administration. So first, let me talk about the gravity method. So first, if we are considering this method, we take a lutetium dotatate vial and we insert a short needle which must not touch the solution inside the vial, and that is attached to through a clamp to the sodium chloride solution, which is of now 0.9% in 500 ml, and it can be controlled using clamps. And we do not allow this sterile sodium chloride to flow into the vial prior to the start of the infusion. Next, we insert the long needle into the vial, and this long needle must touch and be secured to the bottom of the vial during the entire infusion process. And this long needle is attached to tubing, and with the help of a catheter inserted into the patient. So now that the gravity method is clear, it might be helpful to also get to know about the second method, which is the manually, if we manually draw out the contents of the vial into a syringe and then use a shielded automated syringe pump to administer the agent to the patient. So here we see that that amino acid in solutions are also infused into the patient along with the lutetium dotatate and NaCl. So what is the reason? So lutetium dotatate is administered over 30 minutes, which is followed by a 10 to 20 minute infusion of this saline solution, which is to decrease the residual of the lutetium dotatate in the body. And the amino acids, these are used to reduce the residence time of lutetium dotatate in the patient's kidneys, thereby reducing the radiation toxicity to the kidneys. and the only two key amino acids which are used are lysine and arginine. So when purer formulations of only these two amino acids are infused with a lower osmolality solution, this markedly reduces the nausea associated with their infusion. Now that the administration procedure is clear, let me move forward to the mechanism of action. So here in front of you is a schematic of a rough schematic of what happens. So if we see this circle, this depicts a cancer cell which is expressing SSTRs, and we have our compound which is lutetium dotatate, and that is binding specifically to its receptor, and this is further internalized into the cancer. So this leads to the decay of lutetium, leading to cell damage. Further, let me explain to you how this happens. So when lutetium dotatate, which is showing high binding affinity for SSTR2, it is bound to these malignant cells which are showing an upregulation of SSTR2, and upon binding to the receptor, this radiolabeled peptide becomes internalized into the cell, and we see a particle emission of lutetium 177 via Auger electrons and conversion electrons, and this induces DNA single and double strand breaks, with the double strand breaks actually leading to the cell death of the tumor and its SSTR positive lesions. So this is working according to the particle emission of Auger electrons in the surrounding, which is causing the primarily double strand breaks, and double strand breaks of DNA is leading to tumor cell death. Next, what happens is we also see an effect called the crossfire effect, which is defined as like due to the range of these particles in comparison to the overall diameter of the cell, the emitted particles tend to traverse several cells, which can be ranged from 10 to 1000. So this is also causing a possible destruction of multiple cells in the vicinity of this tracer accumulating cell. So this phenomenon can be referred to as radiation-induced bystander effect, where the cells that have not been directly exposed to ionizing radiation but are in the neighborhood of such cells behave as if they have been directly hit. So we tend to not only be able to kill the active cells, cancerous cells which are present, along with it the plausible malignant cells which might tend to become cancerous in the future for the patient. Those cells also undergo possible destruction due to the radiation induced bystander effect. And we see that 4 hours post administration of lutetium dotatate, physiological uptake is seen in kidneys, tumoral lesions of course, and in the liver, in the spleen, and in some patients also pituitary gland and the thyroid also show additional uptake. Moving forward to contraindications, the first, let me focus upon the absolute contraindications, which are pregnancy or even ongoing lactation, and the patients with severe hepatic impairment should not be included into undergoing this therapy, along with severe cardiac impairment, patients having severe cardiac impairment, and of course consent is the most important thing over here. If the patient family or the patient is unable to comprehend or understand the entire therapy, then that patient is not made to undergo PRRT. As well as we must have significant sites of active diseases, or else if those sites lack SSTR expression, then that doesn't confirm that those are NETs. And we can confirm this by first undergoing making the patient undergo an FDG scan, which is the um, the usual PET scan. If the patient is not it's not showing primarily glucose uptake, then we can move for make the patient. If the patient is also showing other symptoms of NET or showing some history related to NET, then the patient can be moved forward for undergoing a gallium-68 DOTA scan. But if we see that these sites of active disease are lacking SSTR expression, which means that the tumors have undergone dedifferentiation and have now been actively taking up glucose, then that can be confirmed by an FDG PET scan. So these patients are also not likable for undergoing a PRRT, because they will not be responding to the therapy. Along with that, if a patient is having impaired hematologic functioning or severe renal impairment, then these patients should never be included for undergoing PRRT. Coming to the side effects, like I mentioned before, are nephrotoxicity and myelotoxicity. So coming to nephrotoxicity, which is actually a progressive chronic renal disease where the glomerular cells are relatively radiosensitive and are not able to regenerate. So any sort of irradiation to these renal cells is actually going to cause more trouble than actual use. So these are primarily related to the very rapid clearance of the small radiopeptides that are filtered through the glomeruli and are reabsorbed by the tubular cells, with subsequent retention in the interstitium, which is resulting in an excessive renal irradiation to the glomeruli, which is unwanted. So this is leading to nephrotoxicity, and here comes the role of the amino acid infusion for the purpose of renal protection. So with this slow infusion of this L-lysine and L-arginine, if when administered, it can be before, during, or after the application of the radiopharmaceutical, then this can competitively inhibit the reabsorption of these SSTR specific ligands, SSTR specific compounds, which are absorbed by these renal cells. And myelotoxicity, if you want to talk about that, then it is the acute or the subacute bone marrow toxicity, with patients having baseline nephrotoxicity tend to suffer from more hematologic toxicity after the therapy. And the prolonged circulation time of lutetium 177 labeled PRRT, along with the poor renal function, can actually increase the toxicity to the bone marrow. So the following classifies tumor response according to different categories set by World Health Organization criteria. And firstly, let me explain what is complete response. It is defined as the complete disappearance of all recognizable tumor in the liver, followed by partial response, which is PR, and it is defined as the reduction of tumor size with being equal to or more than 50%. And the minimal response is defined as a reduction of less than 50%, but more than and equal to 25% reduction in the tumor size. Now, coming to the disease type, it can be stable or progressive. With stable disease defined as a reduction of the reduction of the progression of the disease, anyone, so of at least less than 25%, with progressive disease being defined as an increase of more than or equal to 25% in tumor size over that measured at the time of maximal tumor shrinkage, or as the appearance of new lesions occur. This schematic here shows the design of the NETTER trial, which is Neuroendocrine Tumours Therapy Trial. This was a randomized phase 3 trial called NETTER-1 and involved patients with advanced SSTR positive and G1 or G2, which is grade 1 or grade 2 midgut NETs, which were inoperable tumors and was seen to be progressive under the use of 30 milligrams of octreotide long-acting release. The eligibility was for adult participants with biopsy-proven low-grade metastatic or locally advanced midgut NETs that was inoperable and radiographically progressive with the standard dose of 30 to 40 milligram octreotide therapy. So the confirmation of SSTR expression was based on planar indium 111 DTP octreotide imaging, because gallium-68 dotatate PET was not available in most centers at that time. So the aim of the study was to evaluate the efficacy and safety of lutetium dotatate. We see the two study groups had one administered with 4 doses of 200 millicurie of lutetium-177 dotatate every 8 weeks, along with 30 milligrams of octreotide, while the other group, which is the control group, was administered exclusively with 60 milligrams of octreotide long-acting release every 4 weeks, and no placebo was provided for the control group. And a progression-free survival status and response assessment was done according to RECIST, which is Response Evaluation Criteria in Solid Tumors, and were recorded every 12 weeks. The study was aimed with a 5 years follow-up, and the primary endpoint of this trial was progression-free survival, which means that the trial would end when no patient is shown to have a progressive disease. In total, we see that 229 patients were enrolled in this study, with 116 being in the experimental arm and 113 in the control arm. And according to phase 1 and phase 2 clinical trial results, we got to know that lutetium-177 dotatate, which was carried out with only 35 to 50 patients, showed a partial response rate of 10 to 25%. So in the phase 3 trial, we would be expecting better data validating the use of the efficacy of lutetium dotatate. We see over here the results of the NETTER-1 trial with subjects receiving lutetium dotatate had a 79% reduction in risk of progression, which is shown in the first graph, and with an estimated progression-free survival of 40 months, compared with only 8.4 months for the high dose octreotide therapy group. And in addition to improving the progression-free survival, lutetium 177 dotatate PRRT was also associated with a significant quality of life benefit compared with the high dose octreotide LAR. So the quality of life measures showed improvements overall, and specifically for the cases the cases of diarrhea, flushing, and the abdominal pains. So at the time of the initial NETTER-1 publication, actually the overall survival data were not finalized because the 5-year follow-up phase was still ongoing. But with interim analysis, which can be seen in the graph B, which shows it showed that 48% lower risk of death with lutetium dotatate for and with the objective response rate was also significantly different between the two arms, which can be seen in the graph C, and there were 1 complete and 17 partial responses with lutetium dotatate, which is shown as 1% CR, which is complete response, and 12% PR, which is partial response, total giving us a 13% objective response rate for lutetium dotatate group, whereas we see no complete response and 3 partial responses for with high dose octreotide, which is shown as 4% partial response, also which is corresponding to 4% of overall objective response rate for octreotide LAR. And now if we move forward to the clinical efficacy, which is listed over here, which I just mentioned to you. So lutetium 177 dotatate has limited acute toxic effects. Like I mentioned, the side effects that are pretty common when patients undergo this therapy, but lutetium dotatate is seen to show limited acute toxic effects. So the transient nausea and the vomiting due to the metabolic acidosis, which are symptoms linked to the amino acid infusion, which are performed concurrently with PRRT infusion as a renal protective agent. So this is actually pretty common, but it is not so harmful when compared with octreotide LAR alone. So moving forward to the long-term safety of PRRT in patients with NET, here are the 3 main points which are noted as yttrium-90 labeled PRRT was associated with a significantly higher rate of nephrotoxicity, which was 43.9%, and persistent nephrotoxicity of 33.6% when compared with lutetium labeled PRRT, which only showed a 25.5% occurrence for nephrotoxicity and 13.4% respectively. So this proves that lutetium 177 labeled PRRT is of a more favorable choice because of the lesser occurrence of nephrotoxicity. But when we combine yttrium-90 with lutetium 177 and we have a PRRT which is combining the both these radionuclides, so we can expect that we will be targeting both ranges of we'll be covering both ranges of tumors from small to large, and this was also associated with a higher rate of persistent nephrotoxicity than lutetium 177 labeled PRRT alone. This is primarily due to the higher beta energy emitted by yttrium-90, which might be the reason causing more amount of nephrotoxicity, which is not seen in the case of lutetium-177, and that's why the percentage is only 13.4% compared to 23.6% for a combined therapy. But the prevalence of acute myelotoxicity is seen less in patients treated with lutetium labeled PRRT, which was 2 to 3%, compared to yttrium-90 labeled PRRT, which was 10 to 13%. So this truly proves in front of us the efficacy of lutetium over yttrium-90 for the purpose of PRRT, as it has lesser chances of occurrence of both nephrotoxicity and myelotoxicity in a patient. So this makes it clear in front of us the safety of using lutetium 177 over yttrium-90 for patients undergoing PRRT. So to conclude now, I would like to say that in the future, PRRT will be extended likely be extended to many other tumor types as mentioned before. So thank you for staying connected with Positron Academy. Please do subscribe to the channel for more informative videos and keep encouraging us. Do share with your friends and classmates, and let us all learn together in this process. Thank you.